Hello, everyone, and thanks a lot to the organizers for the invitation. It's a real joy to be here. And I'm going to share recent data from our lab about, we do attempt to ask rodents whether they do something like a hallucination and how this may be changed under psilocybin and how psilocybin changes neural activity more in general. So, maybe it's always like a startup thing. I'm pressing hard, like I'm pressing really hard. Okay, now it's coming. So, of course, we know that psychedelics change perception. They can cause hallucinations. And I like this quote from Aldous Huxley describing his experience with mescaline in the famous book, The Doors of Perception. Visual impressions are greatly intensified, and the eye recovers some of the perceptual innocence of childhood when the sensum was not immediately and automatically subordinated to the concept. And why do I like it? Because it speaks for a potential cognitive mechanism regarding how we integrate sensory information in priors, and this is really what is the foundation of this kind of research. How do we measure more systematically ask humans whether they hallucinate or not? Of course, there's the famous 11-dimensional or the states of consciousness inventory, and we have 3. And for this, I have to also press long, right? And then it comes up or not. Okay, then I've highlighted in red like these 3 dimensions here that capture changes in perception, including hallucinations, and those very well correlate with the dose of psychedelics and a very consistent, is a relatively consistent feature of psychedelic drug action.
Which causes you to see patterns, objects,
and
faces embedded within text.
Can I somehow change the volume, or can you guys mute it? Just muting. Thank you. All right. So I found this little clip on YouTube, and I found this like a nice visualization of like a medium dose psychedelic effect. So I wanted to give some kind of, again, a reminder and an intuition about the perceptual changes that we may see here. So we're looking at just a rock, but as you can see, there is the formation of patterns that is called apophenia. We tend to see patterns in unrelated things. And there's a specific version of seeing faces, which is called pareidolia. I'm going playback. The looping doesn't work somehow. Okay. So we can characterize hallucinations in a bit more detail as like a perception of something without a causative stimulus in the environment that has a subjective quality that is close or equal to a veridical perception, and which is outside of our voluntary control and which occurs when we're awake. So most of these characteristics would, for example, be happening here, perhaps not fully close to a veridical perception, but we see this here rather on a continuum. But the problem remains, how would we ever hope to study this in a translation model or study this in animals? Because hallucinations, as you've seen, are highly subjective. And here we start from a computational view of the brain, and we formalize the computational processes that may give rise to hallucinations in the framework of Bayesian perception and predictive processing. We use this computational framework to link specific neural circuits with overt behaviors that we can see in both humans and animals. And this allows us to then go into the animal model and investigate the same computational processes, even if we can't directly ask and get some kind of verbal or explicit reports, obviously, from our rodent subjects. And with this, I'm going to talk about 3 things here. The first one, which is going to be the focus, is how we are studying hallucinations in animal models, or hallucination-like perception in animal models, as part of an ongoing perceptual inference process. And then secondly, I'll show recent data that psilocybin causes hallucination-like perception in rats. And finally, share some pilot data of psilocybin's impact on neural activity in the mouse brain. Okay, so the first approach, this was developed together in my postdoc time, spearheaded by Katarina Schmack, who is now at the Crick Institute, and my postdoc advisor, Adam Kepich, at Washington University. So the general framework is that of a Bayesian perception or predictive processing. The idea is, of course, that our perception is not just a one-to-one mapping of our sense data, but a combination of sensory information and expectations, or priors in the language of Bayesian perception. So for example, you may see an image like here on the left with a somewhat ambiguous sensory stimulus, and in the context of a bathroom, you may just see a hairdryer. but in the context, say, of a dark alley in a city night, you may actually see something quite different. And this prior expectation here is context dependent, and we can potentially control it. And this allows us to systematically study the process of integrating prior expectation and sensory evidence. Regarding hallucinations or also psychedelics, I would say, coarsely speaking, there are two main theories about how such hallucinations may arise. And one is like the relaxed prior idea, and this you've heard about before. It's in the psychedelic field, the somewhat more well-known idea in which you reduce the influence of the priors and have like a relative increase of bottom-up sensory information, which may cause false or altered percepts. The other model is the opposite, which is actually prominent, say, in schizophrenia and psychosis research, where the idea is that you have an increase, strength in prior, so a relative decrease of the sensory evidence, and this causes you to perceive things which are not there. I want to note that those are not necessarily mutually exclusive. It's about the balance between priors and evidence, and they may be applicable at different levels of the predictive processing hierarchy. These are all questions that we mostly don't know. So here we want to leverage this framework to study the neural basis of psychedelics and of hallucinations across species. So again, now we operationalize hallucinations as false percepts with high confidence. Why high confidence? Because this tells us something about how veridical a percept is that you perceive. And we can also ask about confidence in animals. So how we do this? Now we get into detail into the behavioral task that we employ. This is an auditory detection task in which we have a background white noise. And then we have a brief signal, which is a frequency upsweep signal, something like weep, that is embedded in this white noise. The animals are trained to report signal versus no signal in a two alternative forced choice logic. So there's these pokes. You see like sort of the pink. No, it used to work a little bit. It's down here on this side of the screen, perhaps. Okay, now I found it. Okay, interesting. So we have a left poke and a right poke. In this case, the left poke indicates a signal poke, the right poke, there's no signal poke. So this is a signal trial, okay? We have a series of trials here, and when the animal, the right here, makes a left port choice, it will be rewarded eventually, and if it makes a right word choice, it's incorrect and will not be rewarded. Now there's another twist here to the task, which is that reward delivery is randomly delayed, drawn from an exponential distribution for up to 8 seconds. So what it happens from the animal's perspective, you make a choice, you want to get your water, but you don't know if it will arrive or when it will arrive. So you keep investing time into your choice until there's a self-determined leaving decision when the animal leaves and restarts a trial. And we consider this a time investment into a decision that the animal makes. And we will see that this can serve as a readout of confidence. Okay, so what kind of outcomes can we get here? In terms of a signal trials, there's a standard sort of four outcomes that we can have here from signal detection theory. If a signal was present and the animal chooses signal, we have a hit, it gets rewarded. If the animal chooses no signal, we have a mistrial and there's no reward. Now we also have trials, 50% of trials randomly interleaved that are just no signal trials, there's only noise. In this case, we have false alarms, which are when the animal chooses signal, but there was just noise, okay? These are going to be important. And then there's correct rejections, which are also rewarded when there's no signal and the animal reports no signal. Okay, so then we can go back again to our operationalization. False alarms now, which are when the animal chooses signal when there was no actual signal shown, perceived with high confidence, which will mean a long time investment, and we're going to unpack this in a second. Okay, so the general problem here in animal behavior with these kinds of tasks is how to interpret these false alarms. We do see a baseline false alarm rate of around 10 to 20% or so, but we usually have a very hard time interpreting an error. And this is when confidence now comes in and help us to better interpret what a false alarm may mean. So overall, in a behavioral session, we get a distribution of time investment. This is how long the animal waits in a specific trial. And we get a fairly wide distribution from around like usually 1 or 2 seconds to up to 10 seconds here, in this case peaking at around 6 seconds. But how can we know that in an individual trial, time investment is a behavioral readout of confidence? And for this, we employ standard signal detection theory model and use a statistical definition of confidence. The idea here is that you have a percept which is drawn from the signal-to-noise ratio, the loudness of the signal that is played back in a specific trial. And this percept then is applied to a thresholding mechanism that makes you either perceive or not perceive this signal. And over the course of many trials, this leads to a psychometric curve, and we can use the slope of the psychometric curve to estimate the noise in the system. We then use a statistical definition of confidence as the probability of being correct given the amount of evidence that you have at your disposal, which is your percept. And these then needs to be mapped onto like a specific time investment that may depend on many processes, such as how much you value rating, how effortful rating is, and so on and so forth, that eventually gives rise to the time investment distribution. Now what we can do is, we can ask, now when we employ this model, we can predict what is the optimal time investment in each individual trial, which uses the sensory evidence that is available to the animal. And this will yield a specific set of predictions and test in the data to argue that yes, indeed, time investment is a readout of confidence. And I've plotted some of these predictions here, the first one being the most important one, which is that time investment here predicts the accuracy. So if you see here from 2 to 10 seconds, we predict the full range of the accuracy from around 50% to 100% of performance. In the middle block, you see trials split by correct and error trials. Correct being green. Sorry, there's I think a missing legend here. The green is correct trials and red is the error trials. And even for the same signal-to-noise ratio, the amount of time that the animal invests is predictive of the outcome, whether you made a correct or incorrect choice, which goes back to the statistical notion of confidence. Okay, so equipped with this, now we can finally have a package here, foundation of this kind of behavioral framework that allows us now to look at false alarms with high confidence, that long time investment. In the one study that we did here, spearheaded by Katarina in the lab of Adam Kapich, used this task in mice and also had a human cohort. So there's a human version of this task, which very much looks like the same. So we have humans performing this task, in this case, a web-based version here, whereas the same noise background, there's also upsweep stimulus embedded in the noise. There's a yes-no response, and there's also a confidence report. We get the same 4 types of trial outcomes. We also do see a baseline false alarm rate that is somewhat lower as in the animals, but it's still there. And in this data set, there was a questionnaire quantifying the CAP score, which is how prone to hallucinations you are in your everyday life. There are questions like, do you sometimes hear noises, but you cannot make out the source, or you know that they're not there, and questions like this. And what we found is that this CAP score was correlated with the amount of false alarms that you would do in your task, and especially with the number of high confidence false alarms, which are our model for hallucination-like perception. So this indicates that there's something that we're capturing about hallucination-like perception with our behavioral task. Now, we then reasoned, if this is a good model for hallucination-like perception, it may be a good model for psychedelic-induced changes in perception or hallucinations. So here we did a study in rat where we typically used 1 milligram per kilogram IP injections. And of course, we see all of your general behavior markers. We see an increase in head twitch response and these back muscle contractions, or I think Mikhail called them wet shakes, or I don't know if there's a yet common term in the field. That are somewhat dose dependent, and they're completely absent without the injection of psilocybin or just control injections. We do see a good task engagement with our dose. What you see here is the number of trials that the animal do across time. So typically they spend around 2 to 3 hours in our behavioral chambers where they self-initiate trials. So we just here look at the trial rate of those animals. And what you typically see is like a pause in the beginning where they sit in a corner and don't move much for about 5 to 10 minutes. And this is somewhat dose dependent, as you can see a little bit here with the colored lines. And then they will re-engage in a task. And if you look at the slopes, and also why then we chose 1 milligram per kilogram, because for higher doses, they do the trials quite a bit slower. For 1 milligram per kilogram, which is this brownish, close to the black line, it's almost the same trial rate after this initial pause as without injection or as with PBS injections. So we get decent task engagement, so this allows us to look then at the behavioral performance in this task. Now, the first important party to look at, can we even analyze high confidence in our animals that have a psilocybin injection? So here we're looking at the same signatures that I've shown you before, now in this data set, where we have PBS injections and psilocybin injections, and we see that these typical signatures for confidence are preserved. So time investment predicts accuracy here in the leftmost plot, and also we see the signature that time investment is predictive of whether the animals made a correct or incorrect choice. Again, the legend here is missing. Solid here is correct, and dashed is error choice. It's like here, and then it's here. Okay. But and then it disappears. Okay. What you may see between the middle in black, which is PBS, and the right in pink, which is psilocybin, there's a little decrease of overall time investment. The animals are overall slower. They have longer movement times, as you've heard before. This is a common effect observed. And here they invest a little less time on average. Now we can still do the mapping between time investment and confidence. We ask about the variability of time investment and how informative it is of confidence. And we see that this is largely preserved. We take into account the small shift, which may be interesting in its own right, but is not the focus in the current analysis. Rather, we want to look at the psychometric now with psilocybin. And what we see here is this is a single animal example. We see here all the way the point to the left at like minus infinity are the only no signal trials where there's only noise and no signal. We see our baseline false alarm rate, and we see that this false alarm rate is increased after psilocybin. We see that the hit rate all the way to the right is preserved. They don't make more mistakes for easy signal trials, and we see then there is this gradation here of the effect along the shape of the psychometric curve. We can look particularly at our proxy for hallucination-like perception, the high confidence psychometric curve, and we see that then this effect is even more pronounced. And now this is a big advantage here of the right. So we have done a previous study with mice on this, which is published, not in the context of classical psychedelics. And here, we had to pool all of the animals to really see an effect. And this is now a single animal. We can do multiple sessions. We get thousands of trials. So we see robustly that within a single animal, psilocybin increases the false alarm rate and the percentage of hallucination-like perceptions. Then we did this in our cohort of 11 animals here, and we see that in our cohort, all of those effects hold. And here in the middle on the right side, there's the false alarm plot. False alarm plot here, and the HALIP plot now on the session before drug administration and on the session after drug administration. And you see this increase of false alarms and HALIPs. Here goes up, and then it recovers again in the session afterwards. Here's a plot of the individual animal results. So there's kind of a lot of data here, but so these are 11 animals. Black is always control, and pink is always psilocybin, okay? And we see oftentimes this increase. And because we get a lot of data per animal, we can a little bit look like, how does this look like in individual animals? And we do see a few what could be non-responders, for example, here. Yeah. And we have one animal where it's the opposite. I have no idea why. There's one animal here that is an outlier in that regard. All the other animals point in the same direction, and we have both male and female animals here in this cohort. What you can also look at is at the propensity of having these hallucination-like perceptions across time from injection. Okay, so here we inject at 0, either PBS or psilocybin. And then we look at HALIP. We always have to do this as a running average over many trials, because this only is a number we get from like a couple of trials at the same time. So we use a running average of around 30 trials here, and this is combining animals. So this actually relies on a fair amount of data here in this case. And we see this increase here after around 20 minutes or so. Typically the start engagement around this time or so. And then at around 80, 90 minutes, this effect is gone again, and this matches fairly well, for example, psilocin data in the rodent brain. Okay, we can block this effect with getansrin. So this is, we inject first getansrin and shortly after psilocybin in this case, and we see that the effect is completely gone, and there's even perhaps a slight reduction in hallucination-like perception, which is kind of astonishing, but perhaps makes sense if we are putting the animals already in this conditioned hallucination situation where there may be change percepts in the baseline, which is reflective by this high false alarm rate. It's a little hard to say, but at least the HALIP effect here is gone. Okay, that part was about our behavior model. And now 20 minutes are up, but I just want to give a brief outlook on where we're going with the neuroscience with this. So we are conducting neural recordings now in this task, and we want to get closer to the neurocorrelates of these kinds of behaviors. And in the meantime, we have a large data set in the mouse. And I think it is maybe just nice for you guys to know, because this is going to be published in 10 months. This data set is going to be published, and then everyone can analyze whatever they want to do with this, if they have any ideas of what to look for in this kind of data. So this is an open scope project with my postdoc Roberto, that the Allen Institute then conducts these kinds of experiments where they have a head-fixed mouse and they insert 6 neuropixels at the same time. And then in this case, they record from a single mouse for 2 days. And on the second day, there's a psilocybin injection in the middle of a session. There's a fair number of units here across a couple of dozen of brain areas. And this is also complemented here in a collaboration with Christoph and Irene, who have their own data set where they have either psilocybin or psilocybin injections across yet another number of brain areas here. And what we see is a slight increase in pupil size and no change in running speed, although this will need much more detailed analysis, because running, we do see potential higher order statistics effects in the running, but on the average, running speed doesn't change of these head-fixed mice on a wheel. Slight increase in pupil. And what we then see in terms of the neural activity is mostly a net decrease in activity. Okay, this is like very coarsely. This is also, we have this data set for 1 month. So this is the first coarse analysis here. We see a net inhibition. So here, again, pink is psilocybin, 0 is the time from injection, and black is control here. And then psilocybin here decreases this activity. Okay, this is the difference between pink and black. Now, this is everything combined, yeah, kind of crazy, but just like, okay, this is the net effect. We can split by networks or like coarse regional networks, for example, cortex, thalamus. These both show a decrease in activity. Thalamus is here in red and cortex in like light green. You see this also here, thalamus decrease, cortex decrease. And this is all in regular spiking neurons. We can do this in fast spiking neurons where we also see a net increase. In the interest of time, I wrap up now. We can, in cortex, go into a little more detail and look at the individual cortical areas. We mostly see a decrease with some differences between areas, especially frontal and somatosensory motor areas here show this decrease. The decrease is mostly in deep layers, layer 5 and layer 6 here. And in a few areas, we see an increase in fast spiking putative interneuron activity. And here in some of the frontal cortical areas, which may indicate a shift into excitation-inhibition balance. Okay, and with this, I'm going to wrap up, that I've shown you a framework of how we hope to study something like hallucinations or mini hallucinations across species. We can want to take this quite a bit further, of course, and look more specifically at the weighting of priors versus sensory evidence with standard models of Bayesian perception here, and do the neural recordings to follow up on top-down versus bottom-up predictive processing hypotheses. And we've also seen psilocybin inhibits wide networks, neural networks, including thalamocortical networks, and perhaps shifting the EI balance towards inhibition. Okay, and thanks again for your attention, and happy to discuss this at any time more. And thanks to the lab. Thanks to the lab.